So very good afternoon to each and every one of the participants. Now I am going to. Hello, visible. Okay. Okay, fine. Okay, I am going to talk on screening, cultivation, optimization strategies of microalgae chlorella vulgaris, or particular isolate NTEB09, or for biodiesel production. And this is our output of the two major projects we received from Department of Biotechnology, Government of India. One is Indo UK joint project with the other collaborators from UK, that is Plymouth Marine Laboratory, particularly Dr. Carol Levelin. She is the principal investigator of our collaborative project, and Dan White and Karan Wright, they are the co-investigators. So in our side, Dr. Muralidharan, my one student, now he is a lecturer in our department. He is the co-investigator. Another very big scientist, or I can say that renowned FICO remediation technologist of our country, Dr. V. Subramanian. He also the co-principal investigator of our Indo UK project. So we received nearly 1.5 crores for completing this microalgal biodiesel project work. And another project, again from Department of Biotechnology, Government of India, it is again the network project from National, that is NBRI, National Botanical Research Institute, Dr. M.R. Susila, Dr. S.A. Ranade, and Manjusha Srivastava, they are the collaborators of that project. And now I am going to just briefly explain what is microalgae and cyanobacteria. They are the very minute photosynthetic plant, microscopic plants. It is having all the capabilities of the higher plants, but it is superior than the higher plants in so many aspects. So it grows in all aquatic environment. Wherever moisture and sunlight is there, definitely you can get this growth and establishment of this organism. And they can grow in, they can found, or we can grow this organism in any kind of water, fresh water, marine water, sea water, high pressurized water, waste water, and any moisture we can grow it. And it has a very large, vast potential applications in it. And here you can see this big algal bloom. This is the backside of our University Ladies Hostel sewage. You can see the bloom, and if you see under the microscope in the sample, you can get some beautiful creatures of microalgae and cyanobacteria in it. And why these organisms, microalgae, are more advantageous for almost all the aspects. So one is, as I said, it is biological system harvesting solar energy via photosynthesis. And the most important point is, hello?
Sir, sorry to interrupt, but sir, are you changing the slides? Because we are only seeing the first slide.
No, I'm changing the slide.
So
I'm in the second slide, third slide.
No, sir, that is not visible. We can see the first slide, that is screening, cultivation, and optimization strategies of microalgae chlorella.
Oh, I am in the fourth slide. I changed it, so I'm seeing my slides.
Actually, slides are not for.
Now we see a slide that is called applications. Yes, sir, now we can see the slides being changed.
Okay. This slide, the Indo UK joint project slide, is it visible?
Yes, sir, this is visible.
So, as I said, it is we have two projects.
Visible but not in presentation mode. Please, if you can just.
It is visible in my screen.
Please come back and give that whole window, not only
I'm in a whole window.
Sir, slide show window.
Window other person kill it.
Stop sharing. Okay now. Okay now. Please now. First go now. The next slide was not to burn. No, it's not coming. Design project algae technology. No, it's not coming. Please give entire slide. I mean entire screen. Please give entire screen.
இன்னொன்று போங்க போயிட்டு அந்த விண்டோ போங்க இப்போ ஷேர் பண்ணுங்க அதில் ப்ரெஸ் பண்ணுங்க இப்போ ஷேர் பண்ணுங்க இப்போ ஷேர் பண்ணுங்க
ஷேர் கொடுத்துருங்க இப்போ ஃபர்ஸ்ட் லைட் வந்துடணும். ஓகே
Sir, I guess it's fine because we can see whatever there is in the slide. If you are moving the slides and that is visible, I guess it's fine because the content is visible.
No, after he goes to presentation mode, it doesn't change.
Professor,
He changed it the way he changed before, just few seconds back, the way he was changing.
Please, nobody interfere in the proceeding. Control students will take care of it. Please don't interfere. Sir, you proceed. It is clear.
Full screen. Is it clear now?
Indo UK, please proceed.
Is it next network project on Excel. Yes, sir. So now you can get this slide Indo UK joint project. Is it clear now?
Yeah, it's clear, sir. It's visible.
Okay.
Go ahead.
Okay. So these are all the advantages of microalgae, why it is so important and utilized to all the scientists of the world. So first I said it is a biological solar harvesting system, just like a higher plant. It harvests the solar energy via photosynthesis. And the second important point is non-vascular plant. There's no root, no stem like higher plants. So it has a single unicells, colonies or filaments. So that entire biomass can be harvested. So there will not be any waste, unlike comparing with the agriculture crops. And another most important point is induced to produce commercially valuable compounds such as proteins, carbohydrates, lipids, and pigments. So just like going to the grocery shop and pay money and get the desired product, likewise, we can insect the algae just by manipulating media composition and condition. We can get the desired products. Like biodiesel, for example, in biodiesel, we need the higher quantity of the lipids. For this, grow the algae in a huge biomass, and then you give any kind of stress, you can increase the lipids. Likewise, we standardize some methods to enhance the lipid production during our biodiesel extraction studies. And another most important point is simple cell division. So most of the unicells or filaments, they have the amitotic cell division. That is one cell, just a bacterial fission. One cell becomes 2, 2 becomes 4, 4 becomes 8, like this. There is a sustained biomass production is possible only with microalgae and cyanobacteria. And they can be grown in seawater, brackish water, and so on. The biomass production is also very simple, labor intensive to very sophisticated instruments also. So we can grow in a very larger ratio ponds and fermentation system.
Professor, a slide is still, I think, not moving. Maybe you click the presentation mode of the PPT and try. Just click at the presentation mode.
Presentation mode is there. Yes,
The
is
wider presentation mode. It's clear, but slides are not moving. Maybe you click on the presentation mode of the PPT.
No, sir. He has switched to a single window. That's why it's not changing when he goes to presentation mode.
Okay.
Other,
While changing the slide, please change it manually with the mouse. Sir. By hand, sir.
now it is advantage slide.
Go
So
back and
other option
entire
is
screen.
you
Your
choose entire screen. You
Yes.
choose entire
Okay.
screen presenting. Then whatever you do on the screen can be seen. So that option is
Okay.
right.
The entire screen. So I'm going to entire screen mode and then I'm sharing now. Is it visible?
Yeah. So now, yes.
Now it has come.
A perfect, professor. Please go ahead.
Okay. Okay. So now it is the slide of applications. Is it visible?
Yes, sir. Yes, sir.
Yes.
Okay.
Please go ahead.
Okay. Fine. Okay. Sorry for this.
No, it happens.
Some interruption in the net. Okay. No? So shall I start in from the first slide?
No, if you please continue. Professor.
No, sir, that is not.
Okay.
Please continue from the same slide.
Oh, okay. So coming to the applications, if you see these slides, almost all the products we want, everything is possible with the microalgae and cyanobacteria, particularly in the case of, it is a very important part in primary producers. So without, you just imagine, without these primary producers, without the microalgae or phytoplankton, there will not be any primary consumer, there will not be any secondary consumer, there will not be any fishes, there will not be definitely any fishery population, and also there will not be any fishery ministry. Just imagine this tiny organism, phytoplankton or microalgae, reflects up to the ministerial level. And another characteristic feature is the biofertilizers, single cell protein, for example, the spirulina. It has an amazing quantity of the protein, of nearly 71% of protein is with this, when compared to the mutton or chicken, we can get maximum 22% of the protein in it. When coming to the aqua feed, yes, it's a very good, since it is a primary producer, it is a very excellent aqua feed also. We developed some technology on aqua feed in one organism called Formidium vulturianum, the strain number 30501. We transfer the aqua feed technology to one company at the cost of nearly 50 lakhs. And also this pigmentation present in the microalgae and cyanobacteria, particularly the phycocyanin, phycoerythrin, they are autofluorescence under UV light. We can utilize this in the immunodiagnostic kits as a markers instead of using the radioisotopic labeling or the chemical markers. And it has a very good restriction enzymes also. Because our restriction enzymes like EVA1, for example, Anabina variabilis 1, it is available in the market in very low price because the cost of production is very, very less when compared to the restriction enzymes produced from, bacteria and fungi. And another product is Citroforce. The iron chelators, it's medicinally very important. If any person having the high iron content in the blood, we can use this Citrofor called as diferoxamine. It is a kind of Citrofor. It will reduce the iron content. And this is the biofuel. The present workshop is meant for biofuel. Particularly, I am talking on the biodiesel. I'm going to give you very detailed methodologies on how to produce the biodiesel in subsequent slides. The other applications like phycobilly proteins, as Hannah Rachel says, the phycocyanin and the phycoerythrin, we can make it in decorations also, ice creams and rose milk. Likewise, we can utilize because it is non-toxic, highly nutraceutical value. And nanoparticles, one of my students, Dr. Mubarak Ali is with his participants, he is the expert in our laboratory. He is a nanoparticle specialist. So he developed several types of nanoparticles. If you want, the specialty of the microalgal nanoparticle is, if you want any particular size and particular shape of the nanoparticle, you select a different type of microalgae and cyanobacteria, you can get the different shape. For example, if you want a triangular type of nanoparticle or hexagonal or cubical nanoparticle, you select a different algae, you can get the desired shape and type of the nanoparticles. He published so many papers with this nanoparticle synthesis and with good impact factor journals. Toxins, again, toxins, this is very medicinally very important. When Abdul Kalam visited our laboratory, he insisted us to to maintain the toxic algae or toxic cyanobacterial cysts in our laboratory because for the national security as a bioweapons. Such type of don't think that all the microalgae or cyanobacteria are beneficial. Some very less than 1% of the organisms are toxic organisms also. Now people trying to utilize these toxins for anti-cancerous as well as some medicinal purpose people using. And coming to antibiotics, definitely you are getting a very good quality and quantity of the antibiotic. Particularly, one of my students now he is in US. He published one paper on anti-ESBL producing compounds from one cyanobacterium published in the Nature Group's journal. You know, ESBL standard spectrum beta lactamase enzyme producing pathogenic bacteria. If anyone affected, any kind of antibiotic or combination of antibiotic, if you treat the organism because it has the beta lactamase enzyme, it degrades the antibiotic and utilizes the antibiotic as a nutrient source. So it is very difficult to eradicate this kind of ESBL producers. Luckily, one of my students he got some clues, some product from marine cyanobacteria and fatty acids. You know, fatty acids are also equally important, particularly in the case of biodiesel. And some of the fatty acids like gamma linolenic acid, it is highly valuable, medicinally valuable because it has the important role in contraction, repulsion of the heart muscles. So vitamins, no doubt. We have an amazing quantity of the vitamin in any of the cyanobacteria, algae, particularly the spirulina. It has a huge quantity of the vitamins in it. And bioremediation, no doubt, any kind of effluent if you want to treat, if you use the bacteria, cyanobacteria and the microalgae, definitely you can bioremediate the organism. And one of our very renowned 5-core remediation technologist, Dr. Subhasubramanian, also going to give a lecture in another 2 days in this workshop. So you realize how the algae can be utilized in the case of bioremediation at the large scale. He will explain everything. And anti-fertility compounds, again, in the developing countries like India, we are always in developing country stage because of the explosive population. So if you reduce the population, then there is a chance of reaching the developed country stage because to feed and give all the facilities to the explosive population. For this, one of my colleague, Dr. Tirunala Sundari, she worked on anti-fertility compound cyanobacteria, particularly Oslo species. She got some excellent result, and showing she got some major project from Ministry of Earth Science. Such potentials are there. And the basic and fundamental requirement for initiating any kind of biotechnology, including the biodiesel production. So we have to enumerate and identify the cyanobacterial wealth of our country. Then only we will try to isolate, purify, and develop the germplasm collections. And from this germplasm, we select the suitable strains, suitable microalgae for our biotechnological purposes. So in this way, we surveyed with the headship of my teacher, Dr. G. Subramanian. We surveyed the entire South Indian coastal region, including Andaman, Nicobar, Lakshadweep, and Gulf of Mannar group islands. We surveyed, and then we recorded nearly 240 species from our Indian coastal environments. And as a result of our survey, we developed a jam culture collection at the National Facility for Marine Cyanobacteria. It is a David center established in Bharathasan University. Now this center, all the cultures, marine cyanobacterial or microalgal cultures available in this NFMC can be available to any one of the students or researchers, faculty members of our country. Just to go to the website and collect the specimens for their studies. And thanks to DPT for sanctioning the Indo UK projects, we purchased one plant, that is a molecular taxonomy laboratory, mobile taxonomy laboratory, and in which we developed one small lab in the backside. So we are going to remote places to collect the algal specimens because most of the places are highly polluted. Only the pollution tolerant microalgae or cyanobacteria appear as blooms. So we cannot get the 100% diversity. For that, we are moving to remote places where there is no pollution, and we can get the vast diversity of microalgae and cyanobacteria and try to isolate, purify, and maintain the germplasm collections. So the main purpose of our national center is to establish a germplasm collection of freshwater or marine microalgae and cyanobacteria, and the cultures are available to all the scientists of our country for their subsequent work. And this basic biodiesel surveys are needed for getting the very excellent strain for our purposes, various purposes, including the biodiesel. So our students, particularly he is the student, Mr. Baldev. Now he is a Dr. Baldev. He worked in my Indo UK project. So he surveyed the most with our research team, with Mubarak Ali and other agents. We surveyed frequently the various freshwater bodies and collect the specimens and try to isolate and purify these cultures, germplasm. And this is the record of our cyanobacterial specimens we identified, particularly cyanophyceae or blue-green algae or cyanobacteria. We can see 26 species we recorded from different regions like Tiruchirappalli, Thanjavur, and Chennai. And the chlorophyceae, the green algae, again, nearly 30 different species we identified. And the diatoms, the chlorophyceae members we identified from our freshwater habitats. And the percentage of abundant is more in the case of the green algae than the cyanobacteria and diatoms reported. And these are some of the biodiversity we recorded and captured the micro photographs, particularly the cyanobacteria, different and species, and particularly the green algae. The green algae are very efficient candidates for biodiesel production when compared to the cyanobacteria, because the lipid content is very less in the case of cyanobacteria, whereas it is very higher in microalgae, particularly the chlorella, senitosmus, and the chlorococum, and so on. And again, this is different micro photographs of green algae, like Pediastrum, Clostridium, Cosmerium, Euglena, and so on. We have this diversity in our environment, and particularly diatoms. So diatoms are a very ideal candidate for biodiesel production, because the reserve of food material in the case of diatoms are the lipids. So lipids are needed for biodiesel production, and we can get the very excellent amount of lipids as stored food material in diatoms. And as a result of our freshwater survey, again we established another freshwater microalgal and cyanobacterial germplasm collections in Bharat University as a national repository for microalgae and cyanobacteria. Again, this center is sanctioned by Department of Biotechnology, Government of India. We have a good number of we established. And these are the pure cultures we developed and maintained in our germplasms of NRMCF. And now, because of the COVID period, because of the blockage of our laboratory, most of the species were almost extinct, because it is very difficult to maintain this. Again, we are re-culturing and re-subculturing this all the biodiversity we deposited in our. And you can see this micro photograph of our pure cultures. Not pure culture, they are the unialgal cultures, because it is having symbiotic bacteria. And also the symbiotic bacteria also greatly influenced the biodiesel or lipid productivity. So one of our UK partner, they identified the symbiotic bacteria associated with our chlorella. Some of the bacteria is influenced by using the flow cytometry and other sophisticated techniques. They identified nearly 6 different bacteria associated with the chlorella are greatly influenced the lipid productivity or lipid content in the chlorella. And again, these are all some of the culture collections we are maintaining in our germplasm, and this is the confocal microscopic view as well as the bright field microscopic view of these organisms we are maintaining, and some of the other it is continuation of the other cultures. We have nearly 30 different strains micro photographed. And here you can see one special strain called Barticoccus brownii. These organisms having the lipid content is oozing out from this you can see this oozing out of lipids from the tissues. So this organism is very potential in the case of biodiesel production. Coming to the actual microalgal biodiesel, so how we develop the microalgal biodiesel from the scratch. So, this is some technical information about the biodiesel. It is really the alternative fuel for diesel engines. We can make the biodiesel from vegetable oils, algal oil, or animal fats, and it has some positive points like lower, smoke emissions, high flux point, safer and biodegradable, essential, non-toxic, and chemically biodiesel molecules are monoalkyl esters produced usually from the triglyceride esters. For example, if you see this, the triglycerides are nothing but the algal oil or algal lipids. By trans-esterification process, adding the solvent as well as the catalyst, we can make the fatty acid methyl esters. That is nothing but the biodiesel, and glycerol is the co-product of the biodiesel production. And this is this data is available in literature. If you compare the different bioresources from castor, sunflower, palm, coconut, and so on, here we can get the maximum of 6,000 liters from the palm, 6,000 liters of oil per hectare area per year. Whereas if you, in the same 1 hectare area per year, if you grow the algae, you may get more than 1 lakh liters of oil. You can get it because it is scientifically proved. For example, if you are constructing 7 raceway ponds, if you inoculate the algae in the first raceway pond, you can harvest in the 7th day. If you inoculate on the 2nd raceway pond, you can harvest on the 8th day. Likewise, first 7 days you are not going to harvest any product. So after 7th day, every day throughout the year you are harvesting the biomass, and then you can extract the lipids. That's why it is called a sustained biomass production is possible only with the algae. That's why it is expected that more than 1 lakh liters of oil per liter per hectare area per year is possible. And what are the steps for biodiesel production? For example, it has major 4 steps. One is the microalgal biomass production. For this, we identified some robust microalgae from the environment, which has a higher biomass and then lipid production, everything, and the tolerance capacity. For example, if a strain, it is if you're isolating a microalgal strain, it is very seasonal and restricted in distribution. It may pose the problem for the round-the-year mass cultivation. Whereas if any strain which are highly robust and versatile and existing throughout the year with a very climatic condition, these strains are the efficient candidate for the round-the-year mass cultivation. For this, selection of the microalgae is very important. And then after this, after the biomass production, extract the lipid. And then after the extraction lipid trans-esterification process, after trans-esterification, we are getting the biodiesel which is in higher quantities. And the first bottleneck is selection of potential strain. As I said, we have to identify the robust strain from the environment, and from this, how to extract the lipid content. So then again, you have to standardize some protocols to extract the maximum lipid contents. So we standardize this different parameters like the breakage of the cell wall and using the different solvent. What is solvent is very important for maximum lipid production. Likewise, we standardize the lipid production. After this, how to produce the biomass and lipids in larger scale. So we need the manipulate the media composition and conditions and so on. And then finally, how to get the maximum fatty acid methyl esters, that is biodiesel. For getting the very huge quantity of the biodiesel, we have to rapid biomass production by manipulating the media composition and condition. Once you get the huge biomass, then another technology you have to develop is give a suitable stress condition. So if you're giving the suitable stress condition, we can enhance the lipid productivity. So we tested with the different parameters, and we found that some amazing quantity of the algal oil or lipids, we can get it by using different technologies. Now, subsequent slide, I'm going to explain in detail. And as I said, the robust and very versatile organisms needed for biodiesel production, we identified the Chlorella vulgaris. The strain is NTEB09. And this is the microphotograph and confocal laser scanning microphotograph of the species. And this is the taxonomic validation. And our species are very good adaptability in almost all the environmental conditions. Open tank cultivation is possible, and it is possible to develop generate a huge biomass, and also we can generate or develop a huge lipid content in it. So such type of wonderful strain we are having in our laboratory, and we did all the work, all the biodiesel work with this restricted with this flora species. And as I said, we standardized the lipid content by using different cell disruption method. For extraction of lipid, first basic point is to break open the cells. So we use different treatments like grinding, that is glass bead beating, micro oven treatment, sonication, osmotic shock, autoclaving, and water bath. Among the different treatment, we found that grinding compared with bead beating, that is glass bead beating, we can get more quantity of the lipid in this Florella vulgaris species. So this is the standardized one. And this part of the work we published in International Journal of Biotechnology for Wellness Industries by what is the method suitable for the cell disruption in the case of biodiesel production. And another very important paper we published in Biomass and Bioenergy, it is again for more than 4.5 impact factor journal. That is what is the nutrient stress which enhance the lipid accumulation in floral species for biodiesel production. This is again very good contribution. And this paper is shared with Dr. Zafar Ali for the subsequent familiarizing this work. And we optimize the solvents also for extracting the lipid. We used the different polar and non-polar solvents we used. Among this, the chloroform methanol, that is 2:1 ratio, we used, which is useful for extracting the high lipid content from the algal biomass. So this is again a standardized protocol we have. And then the TBT, the Task Force Committee, recommends to follow the Folds method for the lipid extraction uniformly. All the PIs at the time, nearly 17 PIs received the algal biodiesel production. They recommend this methodology for lipid extraction uniformly so that we can get the uniform results. And this is a slide shows the lipid content and productivity. And we found that the species, the same strain, NTEB, showed lipid content about nearly 25.8 in total dry cell weight among all the tested microalgae. This is again the second one is lipid productivity in mg per liter per day among all the tested microalgae. In all cases, our selected strain is showing the higher content as well as the higher productivity. And this is the overall data among the 30 different algal isolates we have. We screened the lipid content and we found that the same species we got a higher quantity of the lipids. And then once we identify that one, we are authenticate the identification by using the molecular fingerprinting and methods using the 18S RNA gene sequencing, and we deposit the sequences in the gene bank also. And now we can make the phylogenetic analysis, and this will be the reference gene for other scientists also. And for standardizing the media composition and condition, we use the RSM. There is a response surface methodology technique to standardize the media composition and conditions. I am very happy that Mr. Vignesh is going to give an elaborate talk on RSM, response surface methodology on the lipid production or optimal conditions. So I'm not going to detail about this. This RSM technology is to identify which nutrient and which proportion, which composition is important for the maximal biomass production. And this is the data of the result of the RSM technique we used using different parameters like pH, urea, potash, superphosphate, and biomass production. And finally, we come to conclude that the urea, superphosphate, potash, and pH, the following concentrations and quantities are optimized thing for the maximal biomass production. So this can be utilized in the in our racewater pond cultivation of nearly 35 liter capacity racewater ponds. And now I'm going to explain how we develop the biodiesel starting from scratch. So now you can see in the first slide, it is the agar plate, which take the chlorella cultures from this single colony, single dot we selected, and then we transfer the single colony in a 10 ml medium, culture medium. And once we get the biomass in 10 ml, we transfer the whole 10 ml medium to with the algae to the 250 ml conical flask. Once again, we are getting the optimal growth. We transfer the entire 250 ml, here this is 250 ml flask. It contains only the 100 ml of the medium. We transfer the entire thing to the half-tin flask. It is a 2.5 liter capacity. So we gradually enhance the biomass from the single colony. Now we are getting the algal biomass of nearly 2.5 liter biomass, and this is the one sodium alginate beads. This is for preservation of our algal culture. So we are maintaining the cultures in different forms like lyophilization and then glycerol stock, including the sodium alginate beads. Different modes we are maintaining these cultures. And from this 2.5 liter half-tin flask, we transfer the content to 250 ml to 25 liter water cans, just enhance the biomass of chlorella. From this 25 liter, we transfer to 125 liter plastic cups to gradually enhance the biomass. And from this, we transfer to the 5,000 liter raceway pond. It is constructed through the Indo UK projects on biodiesel production. This is 5,000 liter raceway pond with the paddle wheel agitation is there. So this is not the actual end product. This is for initial inoculum for the 35,000 liter raceway pond. So this is the 35,000 liter raceway pond. We transfer the 5,000 liter biomass to 35,000 liter raceway pond. Now you can see the huge biomass of the microalgae. Just imagine from single colony, now we can develop in the 35,000 liter raceway pond level. And this is how the paddle wheel and agitation is working in our raceway pond. For example, the agitation is very, very important because if you are not giving this kind of agitation, all the algae are settled in the bottom. They are not actually exposed to sunlight. So it leads to the death phase. So if you are continuously agitating the algae in this paddle wheel, so entire each and every cells of the algae are exposed to sunlight, so that the multiplication and biomass production is very possible. And every day our students are maintaining the level, water level, and then nutrient loss, everything they are maintaining, and then we are getting the sustained biomass. And this is the the structure of our 5,000 liter raceway pond and 35,000 liter raceway pond. And this raceway pond, we can get it in Google Map also, with this geographical location. And this is actual size of the raceway pond we constructed. Here, it has 1 centimeter depth. If it is there, it equals to 1,200 liters in the case of 35,000 liter raceway pond, and 1 centimeter of height. It reflects 150 liters of water in the 5,000 liter raceway pond. So from this we can calculate what is the total centimeter and what will be the total volume. From this volume, what is the the biomass production of the microalgae, we can calculate it. And this is again another data. The efficiency, the microalgae are very efficient CO2 sequestration during their growth. It utilizes the atmospheric carbon dioxide. So here this data, throughout the year, 12 months, students are harvesting the biomass. This is not the standardized harvesting. So whenever our students are free, they have the time to go and harvest the biomass and then try it. So as a result, in 1 year, we are getting nearly more than 100 kilograms of biomass in dry weight. So if you calculate this one, for example, 1 gram of dry weight of microalgae, if you have, it already fixes 1.83 gram of the carbon dioxide from the atmosphere. So by this way, we can calculate, and finally we concluded that nearly 191 kilograms of CO2 is sequestered by our chlorella under the racewater pond cultivation. And another project we received from, we applied and got it from the Department of Biotechnology, that is artificial induction of algal blooms in our racewater pond system. In natural environment, there is an algal bloom. So is it possible to develop the same kind of algal bloom in our racewater pond, because we need the extensive biomass. See, we applied one project and we got nearly 50 lakhs to work out this project. We surveyed the different freshwater algal blooms and then physical-chemical parameters of the environment, everything we studied. And we come to conclude that some of the parameters are positively influenced and some of the parameters are negatively influenced. So we mix all the parameters and mimic the parameters in our racewater pond cultivation system, and finally we are developing the algal blooms in our racewater pond. And this is the one algal bloom we developed during May 4, 2016, and to confirm that we developed the algal bloom. So we have to develop several times with the same technology or ingredient we are using. And we have to prove that if you are using several times and we hope to get the similar kind of algal bloom, then only everyone will believe. And this is the, in May 4, we developed an algal bloom, and this is August 9, 2016, developed another kind of algal bloom. Now we have a technology. Now we are about to patent this technology. The process is going on. That's why I'm not explaining what are the positive factors and what are the negative factors influencing the development of the algal bloom in the racewater pond system. And we have a simple technology, just like filtration technology using the plankton net. We are harvesting the algal biomass, and then as I said, we have to have two technology. One is the rapid biomass production, and the second one is introduce the microalgal lipids. For this, we established a pulsed magnetic field system in the racewater pond system. So it is again a kind of stressor. Dr. Subramaniam, our close associate in our Indo UK project. So with his support and help, we installed the pulse magnetic field in our racewater pond, and it is a kind of stressor. If originally in the algae, what we are using is having nearly 18 to 20% of the lipid. So if you are treating with the pulse magnetic field, we are getting the really very amazing quantity of the lipid production in these cells. So this process, we optimize the medial composition, condition, and what is the frequency of the pulse magnetic field needed for the rapid extraction of the rapid production of the lipids in the algal biomass. And initially we installed nearly 20 channel pulse magnetic field in the racewater pond system, and when we are agitating this one, all the water enters into this all the 20 channels and they get exposed to the pulse magnetic field. And initially we are giving a very less megahertz, nearly 30 megahertz of magnetic flux we applied in this 20 channel, and to our knowledge, initially the first day it is the biomass is like this. So within 5 days, the entire biomass is in a death phase, because even though it is a very low frequency of 300 megahertz, because of 10 channels, it is a very huge stress to the organism. It goes to the death field. So again, we this channel is pulse 20 channel is not good for this biodiesel production or the growth of the microalgae. Then instead of 20 channels, we developed a single channel pulse magnetic field, and just to develop this one, and then we install this one. So entire water is moving in this pulse magnetic field is within this the tube, as well as in the surrounding region of the tube, the hollow tube. So that's why the entire organism is exposed to pulse magnetic field. We got some really promising results when you are using the pulse magnetic field. For example, so we initially tested with different magnetic field densities like 500, 600, 700 like this. Among the different frequency, the 700 frequency, we got nearly 55.2% of the lipid. Originally, it was a maximum of 20%. of lipid in chlorella. In pulse magnetic field, we got nearly 55.2% of lipid. It is really the first of its kind. We reported that too on the second day itself. So then we feel that it is a very good technology we developed now. This work recently it is accepted in Chemosphere journal. Now we are waiting for its publication. So this is a very good technology we developed using the pulse magnetic field first time in our country. And this is once we got the biomass, so we have to harvest and develop the drying techniques also. So initially the algal biomass is when we are collecting in a plankton net, it is highly liquefied. Then we have to get rid of the water, and we need some thick consistency. Then only we can make this kind of manual noodle-like thing, and then dry. So if you are trying with the manual noodle preparation, you cannot see the uniform thickened regions. Some thickened and thinner regions are there. And drying is also taking in different periods because the thinner regions are very fastly dried and the thicker regions are prolonged drying, so that some of the bacterial population in it degrades the lipids also. And this is some bottleneck in this. So for this, there are two instruments are needed. One is, the continuous centrifuge for this 35,000 liter raceway pond. We have to purchase one continuous centrifuge. It is approximately costed about nearly 25 lakhs. And a dryer, solar electrical dryer, it is again 10 lakhs is needed. Likewise, nearly 35 lakhs worth of instruments are required for harvesting and drying. So instead of this 35 lakhs spending, so we developed a very, very simple technology, just spending 1,600 to 1,700 rupees. We developed one simple technology. We filed the technology in a patent also. And this is called the frame ooze out technique. That is food techniques. The name is, formulated by my student, Dr. Mubarak Ali, because it is a frame ooze out technique of drying. We collect we harvest the entire biomass. It is totally liquefied, and then we make the metallic frames and then pour it. And during the semi-solid condition, we make some cuts, cubical cuts. During their continuous drying, it dried and sinks, so it automatically comes out. So this is called the frame ooze out technology. Whereas if you are near to a noodle-like thing, so you have to scrap it and then we can damage the glass also. So here you can see the uniform thickness. So again, this is the very ideal technology. We developed this technology by mere spending of 1,600 to 1,700 rupees. This technology we filed the patent. It is in the patent catalog. Now there are so many queries. We submitted the queries. Now we are waiting for the grant of the technology of frame ooze out technique. Then we go for using the hand mill and make it a powder. From this powder, we are doing some transesterification, and you can develop the biodiesel. So as a result, we are getting the biodiesel. And the transesterification method, there are some catalyst we standardize the catalyst also. Among this sodium, the H2SO4, sulfuric acid is a very ideal catalyst. We are getting the maximum FEM, that is biodiesel yield. And this is overall aspects of our technology. So we develop the algal biomass. From this algal biomass, giving some stress condition, and extract the lipids. From the lipids, by transesterification process, we are making the biodiesel, and glycerol is the core product. And the remaining waste after liquid extraction, remained waste cellular biomass like cell wall and other components of the chlorella cells. We apply. We inoculate in the fermentation system and inoculate with Clostridium ascobutyllicum. We are getting the biobutanol, and Zymomonas mobilis, we are getting the bioethanol. Likewise, so entire biomass can be utilized. There will not be any waste. This is our overall technology we adapted for in the output of our Indo UK project, combined projects. And we tested the GC profiling, our biodiesel having saturated fatty acids, and then monounsaturated fatty acids and polyunsaturated fatty acid contents with the different fatty acid components, lipid components in it. And this is the biodiesel quality. We compare our biodiesel quality with the biodiesel already produced by different authors, from different publications. We compared it, and then so we use this biodiesel, that is mixing with diesel B20, B40, and B50 in the generator engine, and this is the detail of the generator engine. And as a result of all the aspects, we recently published this paper in renewable energy, impact factor of nearly 4.357. And here Dr. Baldev and Mubarak Ali, these are two persons are the key members for developing this technology as well as the publication. And our former vice chancellor, he inaugurated our smart classroom by lightening the lamp. Here you can see the lamp is usually closed lamp, because this lamp is filled with our microalgal biodiesel. And coming to the total output of our work, so we initially surveyed the freshwater microalgal samples from different freshwater locations of Tiruchirappalli, Thanjavur, and Chennai. And 30 green algal cultures were isolated and deposited in our national repository for microalgae and cyanobacteria, that is for freshwater. All the isolates were cultures are assessed for biomass and lipid production. Among this particular Chlorella vulgaris strain, that is NTEB09, found as a potential strain, and we morphologically and molecular biologically characterized using the 18S DNA sequences and deposited them in gene bank also. And we standardized some cell disruption methods like grinding coupled with glass bead beating technique is optimal for the good extraction of lipids. And the solvent system, that is chloroform and methanol 2:1, is best suited for lipid extraction. And then media optimized using RSM technology, so the urea, superphosphate, potash, and then the pH 8.3 is very optimal for the higher biomass content. And a ratio pond capacity of nearly 35,000 liter and 5,000 liter is established and constructed from the DPT funds. And the potential strain Chlorella vulgaris was cultivated in a continuous mode in 35,000 KL, 35,000 liter ratio pond for 1 year from October to September 2016. And growth rate, biomass production, lipid productivity were assessed during summer seasons. In the summer season, the biomass production is seems to be very high. The techno-economic method of large-scale biomass drying technique was developed and demonstrated and named as a FOOT. That patent is filed. And the microalgal biodiesel was produced based on the optimized conditions and characterized by IR, NMR, GC analysis. I'm not showing this results in this presentation. And the biodiesel quality was analyzed from fatty acid profiles and compared with the international standards. The pulse magnetic field was integrated for inducing the lipids in the microalgae. Among these, the various frequencies we used, 700 megahertz for 4 hours resulted the maximal lipid content. And also for rapid biomass production, the artificial induction of algal bloom using through some DPT project, we achieved, and now we have the technology also. The engine performance and the emissions test was conducted with the three different blends of B1, B40, and B50, along with the commercial diesel. So more than 50% of reduction of hydrocarbon emission, 6% increase in mechanical efficiency by using B50, even at the maximum load of 10 megahertz. So likewise, so we got some interesting results in biodiesel production. And thanks to DPT, as a result of our Indo UK project as well as the national repository we developed, and thanks to DPT for funding this one. And this is the partial building we constructed. This is the proposed building of a second national repository for the microalgae and cyanobacteria established at the Biofuel University. and Dr. Sangeeta Kasturi, she is a director, joint director. She declared open our second national repository in front of Dr. Anand, our Vice Chancellor, and Dr. Sivasubramanian also there in the inaugural function. And this is the infrastructure and the culture facility available in our national repository, and almost all kind of microscopes and all sophisticated instrumentation and pilot scale facility. Everything is available in our national repository. And we have the website also, NRMCF website. In this website, if you go there, you get almost all the details, how to culture, how to identify the organism, all are in a very detailed manner. And also we can we tested the distinguished cyanobacterials and algologists of the world for familiar with the future generation. They don't know who are the pioneer in algology. That's why with a great difficulty we secured the photographs of algologists of our world, and we tested it here. Dr. B. Krishnamurthy sir, Dr. G.S. Venkatraman, my Guruji, Dr. G. Subramanian, Dr. Anand, and Borovitska, Dr. Sivasubramanian, and so on. All are there. Dr. G.G. Boy, Dr. L.V. Venkatraman. So almost all the very famous scientists that we tested in our department, new national repository. And this is the end of my slide. Thank you one and all. If you have any doubts or clarifications, I think I exceeded 10 minutes in my presentation. So far, sorry for that. If you have any doubts, you can, if the organizer permits, you ask. Otherwise, you will send it by email. You will answer it.
Professor, you are on time. You didn't exceed. We only took 5 minutes of your time to delayed start.
Okay.
Yeah. So now the floor is open for questions. Anybody wanted to ask can unmute and make a quick questions. So there is one question, Professor. What is the plausible cellular molecular mechanism of lipid induction by low frequency magnetic field? What is, I mean, he basically want to know what is the understood mechanism that a pulse magnetic field is helping here.
No, actually, if you are giving any kind of stress, definitely this is only the adaptation mechanism. For example, if you are now in India, if you check your lipid content, for example, if it is 100, if you next day you go to the US and check the lipid content, it will be the 120 or 130. So it is the kind of adaptation mechanism to avoid the stress. So the lipid is the first component to secure our body. Likewise, lipid is, if you give any exposed to any stress condition, definitely the lipid content will be high to secure our system. That's why initially in first normal condition, we are getting 18 to 20% of the lipid in the chlorella cell. So if you are giving this stress, we initially tried with a different nutrient efficiency, pH, and also we are adding some marine water in this different stress we tested, and we can hardly improve the lipid production of nearly 3 to 5%, not more than that. Whereas the pulse magnetic field, it is a kind of stress, suitable stress. It enhances the lipid content. So that's all. So it is only defense mechanism. The stress is a defense mechanism to produce the lipid. That's all.
Any other question, please? So, Professor, I have one question. Maybe there is a.
Yes.
In what way the symbiotic organism helps? We hear about the symbiotic culture, and
yes. That's why our collaboration with the US laboratory, our US partner from Plymouth Marine Laboratory, we supplied the unialgal culture of chlorella to the isolated different bacteria from this algae, and then they separately tested the lipid content in combination with each and every bacterial associate, symbiotic associate, and they found that among the 15 different bacterial isolates symbiotically with the algae, only 6 bacteria is symbiotically helping the microalgae for the lipid production. So that's why I don't know what is the real mechanism. They tested with some flow cytometry and other techniques. They recommend the 6 bacterial cultures are helpful for enhancing the lipid productivity. So it is symbiotically associated, that one.
Yeah, so fine. I think I'm looking forward to your publication on this pulse magnetic field enhanced lipid growth. Once it is patent or publication is through, please do share us.
Yes.
And this
Yes,
technique of ooze out frame ooze technique. Very nice work.
Yeah, food technique.
Yeah, yes. So other things are similar, simple questions only. I think about cost effectiveness studies. Someone is asking about whether any cost analysis has been done. I think our microalgae are still not on commercial scale. One technology has to be evolved. Maybe you can just quickly comment on it. And the other speaker is waiting to join.
Okay. So based on what our expertise is, except our pulse magnetic field, in normal condition with some other stress we tested, and that approximately costed more than 300 rupees per liter. And if you are avoiding the transesterification process, and if you are using the thermal liquefaction technology, high pressure and high temperature, if you avoid the solvent as well as the catalyst, we can bring down. So we tested with that one also. We supplied our dry biomass to Aban Industries, headed by Dr. Sendil. So we supplied that culture to them. So he isolated and using the high pressure and high temperature, that is a thermal liquefaction technology, it generated the algal oil. So we calculated that at the time, it comes nearly 68 rupees. So if you are not using this solvent, you can reduce the cost of the biodiesel. So for this,
yeah, fine. I think we are running out of the time. Thank you for your elaborate informations and huge amount of end-to-end work. I'm just repenting that I didn't visit your laboratory so long. So you are so close. Still, I didn't come there. I am looking forward to see you there in the laboratory.
You also visited our laboratory.
Yeah. Fine, Professor. Thank you. I think participant, if any other questions are there, please type it on the chat box. We'll collect and pass it on to the resource person, or you can even email to us, and we can get back to you on the replies and the explanations. So I take this opportunity to thank Professor Thajuddin for his wonderful lecture on the culture of large-scale culture of algae and its production of oil and other value-added products, and the entire processes he has elaborated.